Okay, everybody, we are going to try something different. I am not here today, and we're going to talk a little bit about meiosis. So you should have a packet. Kind of looks like this. Sorry, it says inheritance down the middle. Okay, that's what we're getting out. Okay, so pause if you need to make sure they have that packet, please. Now, we have already talked about mitosis, and mitosis is cell division, and your cells do mitosis whenever you cut yourself or you need to grow. So remember, there's this interphase where your cells are just living their life, and then you have prophase where you've dumped out your sock drawer, and your chromatin, which is that tangled up mess of socks, you straighten them out into nice pieces called chromatids, and then you put the matching socks together, and that makes chromosomes. And then in metaphase, they all line up in the middle because your fishermen, your spindle fibers have pulled them there. And then in anaphase, they all pull apart. And then telophase, you have two nuclei and the cell splits, so you end up with two cells. Well, if you notice, mitosis and meiosis sound a lot alike because they are a lot alike. Mitosis, it makes cells when you need to grow or you've cut yourself, you need to replace them. What we're looking at with meiosis is when egg and sperm are made. You're going to see there's still a prophase, there's still a metaphase, there's still an anaphase, and there's still a telophase. So if we have the same names, the processes have to be pretty much the same. So we are doing cell division still, but we are making egg and sperm. So in your body, you have two different types of cells. Okay? You have what we call body cells, and you have what we call sex cells. Okay? Body cells are basically any cell in your body that's not an egg or a sperm. Okay? Sex cells are your egg and sperm. So if you'll notice on the page that says inheritance going down the middle, okay, you will have body forms, two types of cells, and you'll have a 1 and you will have a 2. Okay? That's what we're filling in here. Okay? The first kind of cell you have is called a somatic cell. Okay? Somatic cells are your body cells. That's basically any kind of cell that's not an egg or a sperm. Okay? And they have 2 of each kind of chromatid. Okay? Basically what that means is they have both parts to the recipe. Remember, when you have a gene is a recipe for a protein. And recipes have 2 parts. You have the ingredients part and you have the instructions part. So if you have 2 of each kind, you have the instructions part and the ingredients part. So these cells can make proteins. They have all the recipe they need. They can make whatever proteins that they need. Okay? Now these pairs are called homologous. Okay? Remember, the root word homo means that they are equal. So homologous means that they are identical to each other. As you can see here, they're twins. Okay? So this will be the one you get from your mom. This will be the one you get from your dad. And you have both. You have the instructions part and you have the ingredients part, or vice versa. So you can make proteins. So these cells are what we call diploid. If you're diploid, that means, sorry, but I'm moving here. If you are diploid, that means you have 2 parts to the recipe. Di means 2. So you have the ingredients part, you have the instructions part. So somatic cells are body cells. These are any cells in your body that are not egg and sperm. So they can make their own proteins. Okay. The other kind of cell you make are gametes, or sex cells. Sex cells are also called gametes. Okay? They only have 1 chromatid. So they have half the recipe. So we say that they are haploid. So egg and sperm only have half the recipe. So you can think of the eggs as having the ingredients part of the recipe and the sperm as having the instructions part of the recipe. That's why egg and sperm had to come together to make you, so you would have both parts of the recipe. So since egg and sperm only have half the recipe, they cannot make proteins. They are born, they're made with all the proteins they're ever going to have. So they use up their proteins, and when they use up their proteins, they can't make more. So as you know, they're going to die. So in humans, our diploid number is 46. Okay? That means that our body cells can make 46 chromosomes. Our haploid number means that our egg and sperm only have 23. So they only have half of what they need. They cannot make proteins. So therefore, we have 2 types of cell division. We have mitosis and we have meiosis. Mitosis we've talked about. Mitosis is what you do when you need to grow, you need to make more muscle longer, you need to make your bones longer, or you've cut yourself. Meiosis is going to make egg and sperm. They're going to make sex cells. So mitosis, we're on the bottom left-hand side of your packet, under two types of cell division. Mitosis produces somatic cells. So on your page where it says somatic, if you want to write body cells, that means any cell in your body that's not an egg or a sperm. You're going to get two daughter cells. They're going to be clones. They're going to be identical to each other. They're diploid. Remember, this means you have two pieces. So you have the instructions and you have the ingredients. So these cells can make their own proteins. Let me move this in a little bit. These cells can make their own proteins. It says it takes place in most body cells. Remember, we talked about how nerves can't do this. And it's needed for growth and repair. So mitosis we've talked about. We did interphase, like I said, it's when the cell's living its normal life. We did prophase, where your chromatin untangles. So it's like you dumping out your sock drawer. Your chromatin untangles to chromatids. So that would be like you laying out all your socks, making them nice and neat. And two chromatids come together to form chromosomes. It's like you putting your socks, your matching socks together. Then in metaphase, they all line up in the middle. And then in anaphase, we pull them apart. Remember, in telophase, we end up with two new cells. So on your packet, down the left-hand side, you should have somatic cells or body cells have two of each kind of chromosome. The pairs of chromosomes are called homologous because they carry the same trait. And these cells are diploid 2N. Sex cells or gametes have only one of each. These cells are haploid. So two types of cell division. Mitosis produces somatic cells. Two daughter cells are identical to the parent. And so now what we're going to focus on today is the top right-hand side. We're going to look at meiosis. Meiosis produces sex cells. This is where you have to make new egg and sperm. Now, ladies, you do meiosis before you're born. Females are born with all the eggs she is ever going to have. So, ladies, you're born with about 400,000 eggs. Now, those eggs are sleeping, and since they're sleeping, they're not using their protein so fast. So they're kind of like in suspended animation. But as you get older, your eggs start to run out of proteins. And they cannot make more. So ladies, your eggs die along the way. So you're born with about 400,000 eggs. By the time you reach the age of 70 or 80, you maybe only have about 10,000 of them left. Because as you get older, your eggs get older. And they're going to die because they can't make proteins. Now, guys, you don't start making sperm until puberty, but then you will make it throughout your life. But they're really small. They can only live about 2 weeks, and then they die and fall apart. Because they can't make new proteins. They're born with all the proteins they're going to have. So top right-hand corner, meiosis produces sex cells. You're still going to have prophase. You're still going to have metaphase, all that stuff. But we're going to do it twice. You're going to see that this divides in half 2 times. We're going to end up with 4 unique daughter cells. On your packet, please write this word heterozygous, because theoretically, every single egg a female makes is different. Every single sperm a male makes is different. Nature likes what's called genetic variation. Nature wants things to be mixed up. Nature wants things to be different. So it doesn't want any sperm to be the same. It doesn't want any eggs to be the same. So we're going to make 4 unique daughter cells. And the cells are haploid. Remember, if you're haploid, that means you only have 1/2 the recipe. So egg and sperm cannot make proteins. When they use up the proteins they're born with, they're going to die. So in females, eggs are produced in the ovaries, and males, sperm are produced in the testes. And on your sheet, you can see the making of eggs is called oogenesis. Remember, the root word oo, O-O, means egg. and genesis means to make or to begin. Okay. Spermatogenesis is the making of sperm. And since eggs have half the recipe and sperm have half the recipe, you have to come together. Okay. And so that's sexual reproduction. Okay. Sexual reproduction involves the production and fusion of gametes. If you don't know the term fusion, that means they come together. So think of the egg as having the ingredient part of the recipe. Think of the sperm as having the instructions part of the recipe. So they have to come together so that your cells can make proteins. And so when egg and sperm come together, okay, when the sperm fertilizes an egg, you get what's called a zygote. And zygotes are diploid. Okay. Haploid plus haploid, 1 plus 1 equals 2. Haploid plus haploid equals diploid. So the egg has half the recipe. The sperm has half the recipe. It made you the zygote, and you have both parts of the recipe. So you can make proteins. Okay. So down the right-hand side, okay, you have meiosis produces sex cells. Parent cells divide 2 times. It produces 4 unique heterozygous daughter cells. You then have in females, eggs are produced in the ovaries. In males, sperm are produced in the testes. Sexual reproduction involves the production and fusion of sex cells. And when a sperm fertilizes an egg, a zygote is formed. Now, when you turn the page, you're going to see a bunch of pictures. You're going to see a bunch of cells. And you'll notice there's interphase. You'll also notice there's a prophase 1, a metaphase 1, anaphase 1, telophase 1, and then what's called interkinesis. And then on the next phase, you'll notice prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Okay. We're going to start on the page that says meiosis 1. So you will see a picture of interphase. Sorry, I know it's dark in here. You'll see a picture of interphase with some lines underneath it. I'm going to tell you what to put in those lines. Okay. So we're doing meiosis. Okay. So why meiosis? Okay. We're going to have, this is going to allow for genetic variation. Nature doesn't want clones. And in all honesty, that's why there's not a lot of identical twins. Nature wants every person to be a little bit different. Okay. Because if we were all exactly the same, one thing could wipe us all out at the same time. So when we make egg and sperm, we're going to make sure that they're all different. Okay. So you're on the page that has the pictures. It says meiosis 1 in top left-hand corner, and you'll see interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis, and what's called interkinesis. Okay. And we're going to fill in the lines with the dots. All right. So with meiosis, we're going to have 2 parts, a 1 and a 2. And you'll see they both have prophase, metaphase, anaphase, and telophase. Okay. We're going to do meiosis 1 first. Okay. This is when it divides. Okay. So on interphase, okay, you've got 2 bullets under interphase. Okay. Right. Bullet 1, the cells grow, and bullet 2, DNA copies. Okay. Now remember, most of interphase is the cell living its normal life. And so, remember, only if it needs to divide will it grow in size and will it copy its DNA. So you see 2 bullets there. The first one you're going to write cells grow and DNA is copied. Okay. Interphase is interphase. There is no difference between mitosis and meiosis. Okay. It's exactly the same. Okay. You then have prophase. Now notice with prophase you have 2 bullets. Some of the bullets did not print, so I'm going to tell you what to write where. With prophase, oops, sorry, don't want to do that yet. With prophase, on your first line, I want you to write chromatin becomes chromatid. Which join to make chromosomes. Okay. Remember, chromatin is your tangled up mess. When you dump your sock drawer out, you spread your socks out. Those are your chromatids. And when you put your two matching chromatids together, you make chromosomes. Same thing happens here. Chromatin becomes chromatid, which join together to form chromosomes. The next line doesn't have a dot, but it needs one. You're going to write nucleus disappears. The nucleus still goes away. Third dot. Centrioles make spindle fibers. Third dot. Centrioles make spindle fibers. Those three things are exactly like mitosis. Okay. We went from chromatin to chromatids to chromosomes. Nucleus disappears, we get spindle fibers. Okay. However, this is where prophase is different from mitosis and meiosis. Because in prophase, you can see you have two extra steps. There should be 5 bullets there. There's 5 lines, 5 bullets. The first 3 bullets are exactly like mitosis, but bullets 4 and 5 are what makes it different. Okay. In mitosis, we stopped with chromosomes. We made chromosomes in mitosis. In meiosis, we're going to bring 2 chromosomes together. Okay. And your fourth bullet there, homologous chromosomes form a tetrad. Okay. Tetra means 4. So notice down here, 2 homologous chromosomes then join together. So we go from here to here. Okay. Sorry, I have my 2 fingers crossed together. They link up. And so we basically have 4 legs. Okay. Well, what this allows these guys to do is something called crossing over. Okay, that's bullet 5. Crossing over is when they swap genes. Okay. Right. So crossing over occurs. And that's what's happening down here in these pictures. This is a chromosome, and this is a chromosome. Okay. Here is a tetrad. We have put 2 chromosomes together, and notice they've swapped some genes. Okay. So now instead of all orange, I'm orange and pink. Instead of all pink, I'm pink and orange. Okay. This is why you're going to get 4 different cells. Because when it's all said and done, I'll just use egg for example. One egg's going to get the piece that's all orange. One egg's going to get the piece that's orange and pink. One egg's going to get the piece that's pink and orange. And one egg's going to get the piece that's all pink. Okay. This is where nature allows our egg and sperm to become different. Okay. This is what genetic recombination is all about. Okay. So up until this point with prophase, everything was the same. The chromatids became chromatids, which formed chromosomes. Nucleus disappeared, spindle fibers showed up. Okay. But this is where it's different. Okay. Instead of just having a chromosome by itself, 2 chromosomes come together. Okay. Homologous chromosomes form what's called a tetrad. Okay. Tetra meaning 4, because there's 4 legs here. Okay. And then they'll do what's called crossing over. Crossing over is when they swap genes. Okay, so it says, sorry about me moving it back and forth. Crossing over occurs when non-sisters exchange segments. Okay, so non-sisters, like if 2 oranges were to change, it's no big deal, they're still orange. If 2 pinks were to change, no big deal, it's still pink. But we'll have an orange and a pink swapping. Okay, that's what makes us all different. So when you make sperm, all of your sperm are different. When you make eggs, all of your eggs are different. Then your next thing is metaphase. And you can see in your picture, things still line up in the middle. But this time, the tetrads line up. Remember in metaphase of meiosis, only chromosomes would line up. That's all we would have in the middle. We would just have chromosomes in the middle. But with meiosis, the tetrads line up. So it's still metaphase, things are still lining up in the middle, but you can see in your picture how we have tetrads lining up. All four line up together. And again, in this picture, you can see where they're swapped. Okay. We had some crossing over here. Right. So metaphase, things still line up in the middle, but this time the tetrads line up. Okay. You can look at your picture for anaphase 1. Anaphase 1, things still pull apart. Right. We still pull apart. Okay. And please add this extra bullet to your page. Okay. This time, though, the chromosomes pull apart. Remember before, we would have only had one leg would be here and one leg would be down here. Okay. So they split apart this way. Sorry. Okay. But now we still have chromosomes. This is going to cause a problem. Because remember, when DNA is a chromosome, it can't work. Okay. And if you remember our little hand symbol, in anaphase, when it's split apart, we made chromatids. Okay. Well, today when we split apart, we still have chromosomes. And this is going to cause a problem. Okay, so make sure you add this bullet to your page. Okay. Homologous pairs separate, and we're still going to have chromosomes. Okay. Notice in your page with telophase 1 and cytokinesis, the cell still splits in half. Okay. The cells still divide. Okay. The nucleus is still going to go back, but now we have a problem because each cell has the original number of chromosomes. Notice I have chromosomes here. Okay. I can't make chromatin. Okay. Remember, in DNA, for a cell's DNA to work, DNA has to tangle back up into chromatin. Well, I have chromosomes today. I can't tangle it back up. Okay. So in telophase, my cells are going to divide, my nucleus is going to come back, my spindle fibers will disappear. But when the cell tries to work, it can't. Okay. And that's what interkinesis is. Okay. Right. With interkinesis, it's like interphase, but I can't do anything with my DNA. Right. So the cells try to work, but it can't. So these cells say, hey, okay, I'm a brand new cell, let's start making proteins. But remember, in order to make proteins, you have to have chromatin. Okay. I'm stuck because I still have chromosomes. So the cell has a choice. It either has to divide again or it's going to die. Okay. It either has to divide again or it's going to die. So, okay, for interphase, you have cells grow, DNA is copied. For prophase 1, remember there's 5 bullets. Okay. One of your bullets talks about chromatin, becoming chromatids, becoming chromosomes. Another bullet talks about prophase, talks about the nucleus disappearing. Another bullet talks about the spindle fibers. And then how your homologous chromosomes link up to form tetrads. And then how you have crossing over. So you should have 5 bullets under prophase 1. In metaphase 1, you have that the tetrads line up at the equator. In anaphase 1, you should have 2 bullets. The homologous pairs separate. You still have chromosomes. In telophase, you have the cells divide, but we have a problem because we still have chromosomes. The cell will not work with chromosomes. The DNA, the cookbook, is locked. Right? We can't make proteins, so if the cell doesn't do something, it's going to die. And interkinesis is where it's figuring that out. So it's like interphase, it's trying to live its normal life, but it can't because there's no chromatin. So the cell says, okay, I have chromosomes, I need to split those chromosomes in half, which is why you have meiosis 2. So you're going to turn your page over and you'll see meiosis 2. You'll see prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Okay. In the margin there, between the 2 groups of cells, put meiosis 2 equals mitosis. Meiosis 2 is the second division, and please write it equals mitosis. So if you remember mitosis, it is exactly the same. In prophase, okay, right, notice the second division is very similar to mitosis. Okay, right. In prophase, sorry, there's supposed to be 2 bullets there. The spindles come back, and the nucleus disappears again. Notice, though, we don't have to make chromosomes because we already have chromosomes. Okay. But the spindle fibers come back. The nucleus is going to disappear again, okay, just like prophase. We've dumped out our sock drawer, but our socks are already paired up. Okay. The spindle fibers are going to come back, and that nucleus is going to disappear, which is exactly what happened in prophase, in mitosis. So the spindles reform, and the nucleus disappears again. Okay. Take a look at metaphase. Take a look at your picture of metaphase. That is the same picture we use for mitosis. So just like mitosis, everything's going to line up in the middle. Okay. Sister chromatids line up at the equator. So once again, we have normal chromosomes like you're used to seeing, just like with your hand gesture. Okay. You line your fingers up. So metaphase 2 is exactly the same as the metaphase you're used to. Anaphase 2 is exactly like the anaphase you're used to. Look at the picture. They're splitting apart. You're pulling your fingers apart. It's exactly the same. Okay. That second line isn't supposed to be there. The second line was supposed to be down under prophase 2. So your sister chromatids separate. Okay. So that's the picture you're used to. Okay. Your fingers are pulled apart. Sister chromatids separate. Okay. But notice this is why we're going to have 4 different cells. Notice this cell is blue and green. This cell is red and yellow. Okay. But this cell has a blue and yellow piece, and this cell has a yellow and blue piece. Okay. So you can see in that picture that none of those cells are going to be the same. This is how come you get 4 different eggs and 4 different sperm. Okay. So anaphase 2 is the anaphase you're used to. The chromatids are going to pull apart. Likewise, you can see in your picture, telophase 2 is what you're already used to. The spindle fibers are going to disappear, the nucleus is going to come back, and our chromatids can curl up to form chromatin again. Okay. Spindle breaks down, nucleus reappears, and you need to add a 3rd line. Chromatid becomes chromatin. Spindle fibers break down, nucleus reappears, chromatids become chromatin. And so in cytokinesis, you're going to divide again. But this time you have 4 cells, and you need to note that every single cell is different, and every single cell is haploid. So you're going to end up with 4 unique haploid cells. Every sperm and egg you make are different. They're not copies of each other. They're not clones, or we would all be twins. And they're haploid. They have half the DNA. They cannot make proteins. So when they use up the proteins they have, they die. Okay. So just to take it back, so for prophase 2, you have the spindles reform, and you've added the line nucleus disappears. For metaphase 2, you have that they pull apart. That's what you're used to. Anaphase 2, you have, excuse me, I think I said they pull apart back here. They line up in the middle, sorry. Here they pull apart. Remember that second line isn't supposed to be there. Then you have telophase 2, you're adding a 3rd line. Spindle breaks down, the nucleus appears, chromatids become chromatin, and cytokinesis, the cytoplasm divides. And then down at the bottom, you've written that you end up with 4 unique haploid cells. So every cell is different, and each cell is haploid, which means it has half the DNA.